Recording.
Hi, I'm Daniel Jamieson, the CEO and founder of Biorelate, and today I'm going to be talking to you about how to rank particular drug targets within the lung cancer space using our platform, Galactic AI. So the talk's going to start off with a quick introduction to the company, what we're about, what our technology does. Then I'm going to share a little bit more about how we build knowledge graphs for things like lung cancer, but also a whole range of other diseases using our platform. How to do some interesting things with that, such as ranking lung cancer targets, and then testing some of the scientific validity of those predictions and ranking those targets. So a little bit about the company. We are based around a mission, which is to curate knowledge to advance the world's most promising biomedical innovations. I founded the company in 2014 after some successful research based around using AI and machine learning to explore biomedical research and mine data that's typically very difficult to capture in an automated way. And doing this for the purposes of discovering new therapeutics and helping validate therapeutics from an existing understanding of scientific data. Today, we're a team of 25 individuals. We're still based in Manchester, although fully distributed in the world's current situation. And we have lots of different partners that we're working with across the biopharma space, companies as large as AstraZeneca and small biotech companies like eTherapeutics and Apconix. So our platform is, I guess, what you'd call a high performance computing platform, in that it's based in the cloud. There are hundreds of virtual machines, terabytes of RAM, and essentially it can process millions of text articles very, very quickly, capturing some really useful data that's typically very difficult to do at an automated level. So at present, it processes 32.5 million documents, capturing 6.3 billion concepts, 154 million causal interactions in under 6 hours. So these stats are taken of May, so they might be a bit better now. And then essentially we're serving up these curated results through 3 different products and services. One is an interface that we call the Galactic Web App. Another is through data licensing, such as Galactic Data, and another is through professional services, which is based around helping companies solve their particular drug discovery challenges with our data and our platform. And so we use lots of different document types, things like journals, patents, clinical trials, NIH grants. Essentially, we can put in any documents that we're interested in. We don't differentiate between different sources. Our platform is essentially able to treat each source agnostically, so that we're constantly adding new text sources, and we're doing this on a daily basis. So Galactic is quite comprehensive in that it's updating all of this stuff routinely, and we're increasing the breadth of those sources on a regular basis. So when I talk about annotating concepts and annotating 6.3 billion concepts, I mean really the types of entities and biological entities that we're really interested in biomedical research. So things like genes, proteins, diseases, phenotypes, chemicals, all of those concepts which you mentioned in research, which typically are mentioned in lots of different ways and are very important for us to capture and to understand. And so we derive all of these different concepts from various different ontologies and proprietary vocabularies that we've built at Biorelate, as well as ones which are the gold standard and are available for everybody to use. We're constantly adding new ontologies and vocabularies, and we're also building ones to improve and clean up the state of some of the ones that are out there today. What we then do, which I think is really unique and quite challenging from a computational perspective, is to capture causal relationships between these different biomedical entities, and we capture these in a very contextual way. So for something like interleukin-6, which is increasing the expression of STAT3 and its activation, the causal relationship there is a directed relationship between IL-6 and STAT3, and our software is automatically able to capture these causal relationships. And it's also able to capture these in context. So you have some of the themes mentioned around that, which are cardiac fibroblasts, atrial inflammation, and fibrosis formation. This context is really important for helping us understand how these different causal interactions have been documented as evidence. We essentially capture these causal relationships between various different entity types, which are important for building knowledge bases. So things like proteins, cells, chemicals, phenotypes, and diseases. And so for each of these individual mentions of an interaction, they're given a confidence score, and then they're aggregated together to form distinct causal interactions. And together we capture 18.2 million distinct causal interactions across the documents that we're currently ingesting in Galactic AI. As I mentioned, these are captured in context. And so if we look at some of the data points, which are essentially in context data points for each of those causal interactions, we have over 1.1 billion as of May earlier this year. Each of these concepts is given what we call a proximity score or a relevance score, which factors in the distance and the sectional weighting of both the causal interaction and the concepts themselves. So essentially then you can use these as really nice ways of selecting causal data that have been associated with these concepts, and you can do that using the proximity score. So now I'm going to walk through a case study of how you can take these components of data to explore some pretty interesting things. And in this case, I'm focusing on lung cancer targets. So in order to build and analyze a lung cancer knowledge graph, there are essentially 3 steps you need to go through. The first of those steps in building this knowledge graph is taking our contextual causal interaction data that has been linked to lung cancer. The second step is to then score and rank particular drug targets and training on existing lung cancer target data, so known drugs and their targets that we have in existing databases today, and then to explore the validity of these predicted targets and to understand how useful they are. So that's the path that I'm going to follow with this particular case study. So the first phase in building the knowledge graph is actually selecting the causal interactions. So if we focus just on protein-protein interactions for lung cancer, we find there are around 202,000 protein-protein. And so what you can see there is a very typically messy knowledge graph for lung cancer. It's not actually a representation of the data for lung cancer, but it is it's just a random messy network, which they all tend to look like. The causal interactions that have been selected for lung cancer have come from essentially taking 54 related child concepts from the disease ontology for lung cancer. And each of these causal interactions is a distinct causal interaction. We find 71,000 of them and 7,800 distinct proteins within it. And it is a very well connected graph. So 96% of those proteins are actually in the main graph of that data. So the next phase is to map existing known lung cancer drug targets against that knowledge graph. And so if we look at Chembl, which is a nice database for finding drug targets, in fact, there are around 3,000 distinct drugs and 1,400 odd indications on that database. There are 191 that are single protein targets for lung cancer, and 175 of those can be mapped to at least one or more causal interactions within our knowledge graph. And there you can see in that table some of the different indications that are in that Chembl database for lung cancer. So the next phase then is to try to rank potential lung targets within this knowledge graph. And the way that we felt was the best approach to doing this is essentially to treat each existing lung cancer target as a positive prediction. So essentially anything that is in our, anything that's not a known drug target is a negative prediction. And so what we wanted to do then is to score and rank existing targets and essentially build this ranking system on those known targets, so that the classifier is essentially basing these on that known knowledge. And we're doing this across 4 different measures. We're doing it on centrality, which encompasses things like degree, betweenness, page rank as potential measures. Enrichment, which is a way of looking at the significance of the proteins in the lung cancer graph against all of the other interactions in the causal interaction database. So from what you'd expect normally by chance, how many of those interactions are represented in that database and how enriched are they? And then we have a couple of other measures which are based around the relevance scoring and the confidence that the data has been extracted correctly. We call these max relevance and max confidence. And what we're looking to do is to then take all of these measures and find the best combination of these for ranking known lung cancer targets. So if we combine these different scorers, we find our best measure encompasses a combination of max relevance, max confidence, enrichment, and eigenvector centrality, with the average precision of those together being 18%. And again, bear in mind that positive predictions are known lung cancer targets. So for every so-called false positive here, which is the 82% of predictions that weren't correct, they may actually be lung cancer targets. However, we don't know about them yet. So we consider that to be quite a high number, given that fact. If you were to take a standard measure like degree, for example, you would get around 14% average precision for looking at particular known drug targets. So this is quite a leap, and it's the way in which we wanted to score and rank particular targets in this study. So using that scorer, essentially what we see is that for many of the proteins, they have a small score, and that the results follow a power law with intervention scores above 0.06 are potentially very useful. And 11 of the proteins that we find have an exceptionally high score, greater than 0.25, and they are essentially the usual suspects. We have 6 of 11 drug targets for lung cancer that are already known about today. 3 of 11 are drug targets for other indications, not for lung cancer. They do potentially represent drug repurposing opportunities, but however, it's commonly known that these are particularly useful repurposing ops for lung cancer. And a couple of others on there which we kind of know are relevant to lung cancer already. But essentially, this is good because what we're showing here is that we can accurately predict and rank known targets of lung cancer from essentially just taking data that's been mined automatically from our platform and putting it together as a knowledge graph. So the next question we wanted to ask is, okay, so we can predict, we can rank lung cancer targets today. Of those which we're predicting could be a lung cancer target, how can we actually test whether they would be decent predictions for lung cancer targets? So one way to do this is essentially to remove all of the data after a certain date and to see if we can predict targets that were discovered after this point in time. So we did this by removing everything that was published after 2010, which took out 55.1 thousand interactions, leaving 15.7 thousand distinct causal interactions in our knowledge graph. And we then recalculated the scores and measured them against future scores, which we would get from all of the data. And we do actually see here that targets of approved drugs get higher scores than non-targets with a significant p-value using the Mann-Whitney U-test. So it's kind of neat because it's showing you that you can predict future lung cancer targets from data at an earlier time point. Because of the contextual nature of the data that we have, we can also stratify by different lung cancer diseases. So we can compare things like non-small cell lung cancer against small cell lung cancer. And if we do this, we get various different proteins that are ranked to the top of the pile in these different diseases. So you do actually get some fairly broad graphs for each of these diseases too. So 5,000 for non-small cell lung cancer and 32,000 for small cell lung cancer. One of the things that we also did, and I'm going to gloss over some of the detail on this slide here, is essentially we combined our intervention scorer with one of the other scorers that we developed within our platform, which we call protein similarity. And protein similarity, what it does is it measures similarity of proteins based on their position in the whole interaction graph. And it does this both upstream and downstream. We used our upstream measure and we created 2 SVMs to score the similarity of targets against general approved drugs, so all approved drugs, and an SVM against lung cancer specific drugs. And what this enabled us to do was essentially to plot lung cancer target likeness against general target likeness. And this gives you some fairly interesting results, because what it shows you is at the bottom right-hand corner of this particular plot, you can see targets that have a high lung cancer target likeness and a low general target likeness. So it's signaling that these particular targets may be more specific for lung cancer. Those at the top right have a high lung cancer target likeness and a low, and also a high general target likeness, which essentially means that these are probably more generic targets for oncology diseases which we know about. One of the other things that we can do is to combine these two together. So if we combine our intervention score with our protein similarity score, we can then see proteins which have a high intervention score based on our measure derived from the knowledge graph against a high lung cancer target likeness and a low general target likeness. So in this particular plot, the dark blue points represent those with a high intervention score, and where they're plotted on this particular graph represents their target likeness. So again, the dark blue dots in the bottom right-hand corner are those which are potentially most interesting, because they have high intervention score, and they also have high lung cancer target likeness and low general target likeness, which gives us a really nice way of exploring this type of data. So in summary, what we can do is take causal interaction data that's very contextual and use that to build very specific knowledge graphs for further investigation. And we've done a quick study here on lung cancer across different lung cancer diseases, but really you could do this across any measure of context that you're interested in. So it could be a particular phenotype, a particular area of anatomy, a combination of diseases. We can build knowledge graphs under any criteria that we're interested in because of the flexibility of the context that we have. And by doing this, we can also create some quite nice ways of scoring and ranking particular targets using combinations of relevance, confidence, enrichment, which essentially boosts standard measures of centrality. When we focus in on lung cancer, we can predict known lung cancer targets with relatively good accuracy here, and we can capture most of them within our knowledge graph. And we can also show potential repurposing opportunities as well as interesting novel targets. Lung cancer, of course, can be explored in much greater granularity, and using the context that we have here, that gives you the ability to do it. And if we combine these scores with other takes and ways of looking at lung cancer targets, such as protein similarity, we can further stratify interesting target opportunities. So thank you very much.